All right, and our last speaker for this morning, Jason Shohet, is the Associate Professor, Department of Pediatrics, and the Co-Chair for Neuroblastoma Research Program at Texas Children's Cancer Center at Baylor. Let's see if I can pull all this up for you. And they'll be talking to us on characterizations of a novel cancer stem cell subpopulation in neuroblastoma.
Thank you. Is there a pointer up here, or is it still up here?
Here, yes.
Oh. Thank you very much. I want to really extend a great thank you to Giselle for inviting me to present some basic research at a clinical, sort of a clinical meeting. And although she did put me right before lunch, which was kind of mean. So hopefully I can keep your attention while your glucose levels are fading. And I'm not going to talk about a clinical trial or something that's going into clinic, but I am going to talk about something that may have some clinical impact, and that is the identification of a novel cancer stem cell population in neuroblastoma. So today I'm going to try to relatively rapidly go over how we identified neuroblastoma cancer stem cells based on expression of the GCSF receptor. GCSF is a cytokine used to promote expansion of hematopoietic cells. It is also found in a number of different neuronal tissues. And that's the identification, and also go over some of the characterization. We've characterized this subpopulation of neuroblastoma cells both extensively in vivo and in vitro with regarding their tumorigenicity, their capacity for self-renewal, the molecular phenotypes, and how they are similar to normal populations of pluripotent pluripotent normal stem cells. So I don't have to really emphasize to this audience the fact that the majority of neuroblastoma fatalities are due to relapse disease, and it's now thought that in many other types of cancer, relapsed recurrent malignancies are driven by small subpopulations within the tumor that may represent a stem cell-like or tumor-initiating-like population. So the therapeutic hypothesis that motivated our research is that if you can identify a subpopulation of cancer stem cells driving relapse and that perhaps escape initial therapy, that you could target this population one way or another and therefore either reduce or prevent altogether the possibility of the tumor coming back. So this would be, this sort of highlights the hypothesis overall, is that standard chemotherapy misses a drug-resistant stem cell population, and then these cells drive the regrowth of the tumor. An alternative approach to therapeutics would be to use standard chemotherapy or cytoreductive therapy in addition to targeted molecular therapy to kill a stem cell subpopulation. So having been to many different cancer stem cell talks over the last year, I just wanted to really emphasize the importance of defining these populations. So for our purposes, we're calling neuroblastoma cancer stem cells as a subpopulation which can self-renew, which can generate all the different types of cells within the tumor, and shares some features of stemness with other pluripotent populations, including embryonic stem cells and induced pluripotent stem cells. And in addition, I think that any cell of origin study, any cancer stem cell study should emphasize the fact that cancer is derived from a normal tissue and should reflect the cell of origin of that normal tissue. So the GCSF receptor, so why did we decide to look at it? Mostly because of a postdoc in our laboratory who just would not give up. She presented data to me showing clearly that the GCSF receptor is expressed on neural stem cell populations. It's a ligand that promotes survival and expansion of neural stem cells in several different studies. It's a transmembrane receptor that signals through JAK-STAT3 pathways, which are well known to play a role in other cancer stem cell and normal stem cell populations. So we decided, well, let's take a serious look at looking at the expression of this on neuroblastoma cells. So first thing we did was develop a flow cytometric assay to look at a CD114 using an antibody against CD114. And we looked in neuroblastoma cell lines, we looked in human xenograft tumors, and we looked in primary tumors. And we found that all of the cell lines we looked at, you could clearly find a small subpopulation, less than less than 1% of the total neuroblastoma cells within the tumor, or actually here is cell lines, expressed this surface receptor. And then we looked at Texas Children's. We have the luxury of being able to access primary tumor biopsy specimens at diagnosis and take them directly to the flow cytometer. So this was done all on live cells, and we found that in a panel of samples of patient samples at diagnosis, and these are all primary sections, and these are after 3 cycles of chemotherapy, or one of them is a metastasis. We could uniformly detect this population in all of the samples that we looked at, and it seemed to be enriched after chemotherapy. To summarize this part, we could find CD114 positive cells in human cell lines, mouse, the cell lines derived from the transgenic animal model of neuroblastoma, as well as all the primary human tumors that we've looked at. So we really strongly believe in orthotopic modeling of neuroblastoma. So what we do here is, this is a immunodeficient nude mice, and this is a needle injecting cells into the renal capsule, or just below the renal capsule. And then these mice are stitched up, and we can image the tumors by luciferase or other markers. And we found by doing this, we could compare, if we inject flow cytometry sorted CD114 positive and CD114 negative cells from different cell lines, that primarily we got tumors arising from the CD114 population and not the CD114 negative population. This was very interesting to us. We went on and did some limiting dilution assays, and we found that overall CD114 positive cells were at least 10 times higher in terms of their tumor formation capacity than CD114 negative cells, as shown here. These results were all highly statistically significant. The next thing we did, the color's a little off, but what we did here, I also want to emphasize that we were able to form tumors from 10 cells injected into the renal capsule, which was a little bit difficult to do, but has been very reproducible. So in terms of lineage tracing, to identify, do these cells really drive the tumor formation in vivo, we could do a little tricky experiment. Let me just walk you through this. We could take, using our FACS, our sorted flow cytometry sorted subpopulations, we could take cell lines that are either stably transfected to express a green fluorescent protein or a red fluorescent tag, and then we could mix combinations of either, for instance, in this case, green labeled CD114 positive cells with unlabeled or red labeled CD114 negative cells. And here we took only 500 cells and mixed them with 500 cells, so we had 50% mixtures. And universally, all of the tumors that we found maintain the genetic tag of the CD114 population when we did this. We did this in 2 different cell lines. We did this in NGP, and we did this in SY5Y. Interestingly, when you look at those tumors, you go back and look at the subpopulation, again, primarily the tumor is CD114 negative, and it's maintained a small, low population of CD114 positive cells, suggesting that suggesting that these cells self-renew, and also by lineage tracing, they tend to drive tumorigenesis in vivo. Importantly, all of our assays, we really rely on the in vivo tumorigenicity for defining a stem cell population. Just want to emphasize a little bit of our clinical findings. We've done more samples than this now, but we've always found that after in biopsy specimens or resections that we've looked at after chemotherapy, there's a 5 to 10-fold enrichment of the CD114 population. This was found in, we can continue to find this trend. We redid this experiment using our xenograft model, so we set up orthotopic xenografts again and treated them with chemotherapy for 3 cycles. And then we took the sample, compared the CD114 population at the beginning here when we injected them, and then what we got out of the tumor. And we could show again that when you give chemotherapy, you significantly enrich for the CD114 population in the xenografts. So does this subpopulation within the tumor meet all of our criteria for cancer stem cells? And to get this, we really had to rigorously define our definitions and compare this to what's been published about cancer stem cells and other tumors to get this into the literature. So we can purify them directly from all of our patients. And importantly, there's no cell culture involved here. There's no neurosphere assay. There's no extensive ex vivo expansion of any subpopulation, which clearly alters the gene expression of neuroblastoma cells. We showed that these tumors, that in vivo tumors are preferentially derived from these cells, and they're highly tumorigenic, at least 10 times more. And we're repeating this in multiple cell lines to see just how much tumorigenicity can be ascribed to this subpopulation. Importantly, we find them in all sources of human and transgenic neuroblastoma, so it's interesting that they're also found in the NMIC transgenic model of neuroblastoma and all of the primary human tumors, as I emphasized. So in terms of the molecular characterization of this subpopulation, we've sort of gone on the hypothesis that a stem cell from various sources is going to share some of the molecular phenotypic characterizations of normal stem cells. And fortunately, there's a lot of molecular data available now on embryonic stem cells and induced pluripotent stem cells from reprogramming experiments and regenerative medicine studies. So at the molecular level, typically, stem cells share a common stemness phenotype, which is manifested by specific gene expression patterns, expressions of microRNAs, epigenetic modifications, changes in cell cycle, and overall signaling controls in the subpopulations. So we looked at, this is just an example of some of our molecular characterization. I don't have really time to go into all of the different stuff. It's going to be in a paper in Cancer Research, hopefully in the next week or two. So we did small RNA sequencing to look at the microRNAs of CD114 positive, CD114 negative cells, and you can see right away, just overall, there's a major difference in expression of microRNAs. And many of these microRNAs are NMIC-regulated microRNAs. And if you look at other papers in the literature looking at how neural stem cells development and the microRNAs, these are embryonic stem cells that have been driven to become neural stem cells and then differentiated into neurons. And if you look at the microRNAs upregulated in our neuroblastoma stem cell population, you can see that many of these microRNAs are also upregulated in neural stem cells driven from embryonic stem cells. So that's a very nice correlation. The other thing we've looked at is in terms of the neural crest cell of origin of these cells. This is qPCR data. We looked at many different pathways, and one of to try to see if this stem cell population is of an earlier origin than the typical neural crest cells within the tumor, and we did find that. The neural crest that people talk about as the cell of origin for neuroblastoma is a very complicated sort of transient cell population as children are developing in utero, and then at birth it continues to develop. And one of the reasons why neuroblastoma is found only in children is that neural crest really shouldn't exist after 1 or 2 years of age, or in the mice after 2 or 3 months. So we found that in particular CD114 positive cells express many of the markers of neural ectoderm, which is the precursor to neural crest. So we found not only are they more tumorigenic, but they also sort of fit the developmental pathway of neuroblastoma. So if you think of neuroblastoma as developing from a neural crest, a neural ectoderm which becomes neural crest, and then these cells go on to develop your adrenal gland and the sympathetic nerves and ganglia from which neuroblastoma is derived. We have a model where perhaps MYC, perhaps ALK, perhaps other oncogenes or oncogenic effects is blocking this differentiation and permitting the development of CD114 positive cells, which can then go on and drive the rest of the tumor formation. So that's sort of a brief overview of neuroblastoma cancer stem cells from what we found in our lab, and clearly these are stem-like. We have additional data showing they're epigenetically similar, they're molecularly similar to reprogrammed cells. They're highly enriched in relapsed and post-chemotherapy specimens from patient biopsies at Texas Children's. And the most important thing is the clinical relevance of this. I think we really need to show what's the role. Does GCSF which is given to all of the children who get chemotherapy, does it have a role in the development or the metastasis of neuroblastoma? We're developing a GCSF, we've obtained actually a GCSF knockout mouse model, which we're going to try to test this in vivo directly with allografts of neuroblastoma. And then also, can we target the subpopulation with specific drugs or novel therapeutics looking at the molecular pathways that are activated within this subpopulation? And there are many different approaches come to mind. Once if this is really true, then we should be able to get funding to develop antibody-drug conjugates, test this in combination with chemotherapy, see what STAT3 inhibitors or other downstream pathways of the GCSF signaling pathway do to the growth of neuroblastoma. So I just want to acknowledge, by way of acknowledgement, I do about a third of my time is clinical, and I just want to really express my gratification, my gratitude to all of the children who have let us study their tumors and let us take care of them in the hospital, and also my team. Most of this credit work goes to the credit of Danielle Patterson, who actually her last name is Sue now. She's gotten married. She is a very talented surgical resident, actually, who just would not give up on the concept. And it was her dogged annoyance bringing me more data every day saying, oh, look, there's this population. So I kept making her go back, okay, do this control, do this control. And after about 3 months, I kind of bought into it and expanded her efforts to do the in vivo testing. I work closely with Eugene Kim, who's a very talented pediatric surgeon at Texas Children's, and we have a surgical dedicated oncologic surgical program at Texas Children's that provides us with tissue. I've gotten funding from the ACS, Alex's Lemonade Stand, St. Baldrick's, the Gilson Longenbaugh Foundation, and other foundations to support this work. So with that, I'll end and say thank you.